There is a moment, somewhere around the third week of human embryonic development, when something extraordinary happens. A cluster of cells, no larger than a grain of sand, begins to fold, to twist, and then, without any instruction from a nervous system that does not yet exist, it begins to beat. There is no brain to command it. There is no signal from the outside world. The heart starts on its own, and it will not stop for the next 80 or 90 years if everything goes right. That single fact, that the heart initiates its own rhythm, is not a footnote in biology. It is the foundation of everything else that follows. Most people carry a surprisingly incomplete picture of what the heart actually does. It pumps blood. That much is universally understood. But the mechanisms behind that pumping, the elegance of its design, the molecular events that unfold inside its muscle cells with every single beat, these remain largely invisible to the people whose lives depend on them. And what is perhaps most remarkable is that the more closely scientists have examined the heart over the past century, the more unexpected its true nature has turned out to be. The heart is not simply a pump. It is, in a very precise sense, an electrical organ that has learned to move fluid. That distinction matters, because if you think of the heart as a mechanical device, a kind of biological bellows, you miss the most important part of what makes it work and what makes it fail. To understand where that understanding came from, it helps to go back to a time when it did not exist. For most of recorded history, the heart was understood as the seat of emotion, of courage, of the soul itself. Ancient Egyptian physicians believed that the heart was the center of intelligence. The ancient Greeks placed thought and feeling there. Even as late as the 17th century, the relationship between the heart and blood was deeply misunderstood. The dominant view, inherited from the physician Galen, held that blood was continuously produced by the liver and consumed by the body, that it moved outward from the center and simply disappeared. It was the English physician William Harvey who overturned that model in 1628 when he published his demonstration that blood moves in a closed circuit, that the same blood circulates continuously, driven by the heart. Harvey's insight was radical enough that it was rejected by many of his contemporaries. But it was correct, and it gave science the foundation it needed to begin asking better questions. Not just where does the blood go, but how exactly does the heart move it there? The answer begins with anatomy. The human heart is a hollow, muscular organ, roughly the size of a clenched fist, located slightly left of center in the chest. It is divided into 4 chambers, 2 upper chambers called atria and 2 lower chambers called ventricles. The right side of the heart handles one circulation, drawing oxygen-depleted blood from the body and sending it to the lungs. The left side handles the other, receiving oxygen-rich blood from the lungs and sending it out to every tissue in the body. These 2 circuits are separate but simultaneous, driven by the same rhythmic contraction. Between the chambers and at the exits of the ventricles, there are valves, 4 in total, that open and close with each heartbeat. The sound of a heartbeat, that familiar lub-dub, is not the sound of muscle contracting. It is the sound of valves snapping shut. The first sound corresponds to the closure of the mitral and tricuspid valves, separating the atria from the ventricles. The second corresponds to the closure of the aortic and pulmonary valves, preventing blood from flowing backward after the ventricles contract. The heart, in other words, is engineered not just to push blood forward, but to prevent it from going backward. Every beat is a precisely timed sequence of openings and closings. But none of that mechanical action would be possible without the electrical system that coordinates it. And this is where the story of the heart becomes genuinely surprising. Deep within the right atrium, there is a small cluster of specialized cells called the sinoatrial node. It is sometimes described as the natural pacemaker of the heart, but that description barely captures what it actually does. The sinoatrial node generates its own electrical impulse, spontaneously, rhythmically, without any external trigger. roughly 60 to 100 times per minute in a resting adult. This self-generated electrical signal spreads outward across both atria, causing them to contract and push blood down into the ventricles. The signal then reaches a second cluster of cells called the atrioventricular node, located at the junction between the atria and ventricles. Here, something crucial happens. The signal is deliberately delayed. The atrioventricular node slows the electrical impulse by a fraction of a second, giving the ventricles time to fill completely with blood before they contract. This delay is not an accident or an inefficiency. It is a design feature. Without it, the ventricles would contract too early before they are fully loaded, and output would suffer. From the atrioventricular node, the signal travels rapidly down a specialized pathway, the bundle of His, and then fans out into a network of conductive fibers called the Purkinje system, which carries the electrical impulse simultaneously to the walls of both ventricles. The result is a coordinated, synchronized contraction that squeezes blood upward and outward, to the lungs on the right side and to the aorta and the rest of the body on the left. All of this happens in less than a second, and then it happens again and again, without pause, without rest, for as long as the organism is alive. At the cellular level, the mechanism of contraction depends on a molecular event so precise that it borders on the architectural. The muscle cells of the heart, cardiomyocytes, are packed with protein filaments arranged in repeating units called sarcomeres. When the electrical signal reaches a cardiomyocyte, it triggers the release of calcium ions from internal storage structures within the cell. Those calcium ions bind to proteins in the sarcomere, and in doing so, they unlock a molecular motor. Two protein filaments, actin and myosin, begin to slide past each other, shortening the sarcomere. Millions of sarcomeres, shortening simultaneously across billions of cells, produces the coordinated contraction of the whole heart. This process, calcium triggering the sliding of filaments, is called excitation-contraction coupling. It is the molecular translation of an electrical signal into a mechanical event, and because it depends on precise calcium concentrations inside the cell, disturbances in calcium regulation are among the most dangerous things that can happen to the heart. Many cardiac arrhythmias, abnormal heart rhythms, trace their origins to disruptions in this calcium signaling system. The electrical architecture of the heart is more fragile than it appears, and its vulnerability is inseparable from its sophistication. There is another layer to the heart's function that often goes unappreciated: its relationship with the nervous system. The heart can beat on its own. That much has been established, but it does not beat in isolation. The autonomic nervous system, which governs the body's involuntary functions, exerts continuous influence over heart rate and force of contraction. The sympathetic branch accelerates the heart. This is the system activated during stress, exercise, or perceived danger. The parasympathetic branch, delivered primarily through the vagus nerve, slows the heart down. What this means in practice is that the heart is constantly being fine-tuned in real time. During physical exertion, the sympathetic system floods the heart with norepinephrine, binding to receptors on cardiomyocytes and increasing both the rate and the force of contraction. During rest, the vagus nerve releases acetylcholine, which slows the sinoatrial node and gives the heart the opportunity to recover. Heart rate variability, the subtle fluctuations in the interval between beats, is now understood to be a meaningful indicator of how well this autonomic regulation is functioning. A heart that varies its rate appropriately in response to changing conditions is a heart with a healthy nervous system behind it. The coronary circulation adds yet another dimension. The heart, despite being filled with blood for most of each beat cycle, cannot extract oxygen directly from the blood it contains. It must supply itself through a dedicated network of arteries on its outer surface, the coronary arteries, that branch into smaller vessels and penetrate the heart muscle. Crucially, most coronary blood flow occurs during diastole, the relaxation phase between contractions, because during systole, the contraction phase, the squeezing of the heart muscle compresses those vessels and restricts flow. The heart, in other words, feeds itself in the pauses between beats. This anatomical reality makes coronary artery disease so dangerous. When fatty plaques build up inside the coronary arteries, narrowing their diameter, blood flow to the heart muscle becomes restricted. During physical exertion, when demand increases, supply cannot keep up. The result is a characteristic chest pain called angina, a warning signal that the heart's own fuel supply is compromised. If a plaque ruptures and a blood clot forms, blocking flow entirely, the downstream muscle begins to die within minutes. This is a myocardial infarction, a heart attack. And the speed of that cellular death is determined by the gap between oxygen demand and oxygen delivery. Modern cardiology has developed remarkable interventions for this scenario. Clot-dissolving medications, catheters that physically open blocked arteries, stents that hold them open. But the fundamental biology has not changed. The heart is still a high-demand organ that cannot store oxygen, cannot rest, and cannot tolerate interruptions in its fuel supply for more than a few minutes. What is perhaps most striking about the heart, when viewed through this lens, is how closely its vulnerabilities are linked to its design priorities. The heart is built for continuous, reliable output over decades. To achieve this, it runs hot, metabolically speaking, burning oxygen at a rate that leaves no room for error. It sacrifices redundancy for efficiency, and it compensates for that efficiency by building in extraordinary precision at the molecular, cellular, electrical, and anatomical levels simultaneously. Research in the past 2 decades has also challenged some older assumptions about the heart's capacity for self-renewal. For a long time, cardiomyocytes were considered terminally differentiated cells, cells that, once formed, could not be replaced. The adult heart, under this view, was fixed. Damage was permanent. Evidence now suggests that the picture is more nuanced. Some cardiomyocyte turnover does appear to occur in the adult human heart at a slow but measurable rate. Whether that capacity can be meaningfully amplified using stem cell therapies, regenerative medicine, or other approaches remains one of the most actively investigated questions in cardiovascular biology. There is still no consensus on the clinical potential of cardiac regeneration, but the field has moved decisively away from the earlier assumption that no renewal was possible at all. There is also growing recognition that the heart is not merely a passive responder to signals from the brain and nervous system. It sends signals back. The heart contains a dense network of sensory neurons and produces hormones, most notably atrial natriuretic peptide, that regulate fluid balance, kidney function, and blood pressure across the body. Some researchers describe this as a kind of cardiac nervous system, a local network of neurons within the heart that processes information and influences, function independently of the brain. How deep that autonomy goes and what role it plays in the subjective experience of emotion. Why we feel things in our chest, why anxiety has a cardiac quality, remains an open and genuinely fascinating question. What the science ultimately reveals is a system of interlocking design decisions, each one a solution to a specific biological problem, each one creating new constraints that required further solutions. The heart beats electrically because muscle fibers need coordination that chemistry alone cannot provide. It has valves because pressure gradients alone are not reliable enough to prevent backflow. It feeds itself during diastole because systolic compression is unavoidable. It accepts nervous system modulation because the demands of a living body change moment to moment. Every feature is an answer. Every answer creates new questions. There is something genuinely moving, and the word is apt, about a structure that has been beating in the human chest for hundreds of thousands of years, that continues to beat in every living person on Earth right now. and that still holds biological puzzles that science has not fully solved. The heart is the most familiar organ in the body. It is also, in some fundamental ways, one of the least completely understood. What we do know is this: every heartbeat is a coordinated event involving electrical signals, calcium cascades, protein machinery, valve mechanics, and vascular pressure, all synchronized to a rhythm that the heart itself generates and the brain only modulates. It is not a simple pump. It is a self-organizing, self-regulating, self-powered engine embedded in the center of a living system. An engine that carries within it the full complexity of what it means to be alive. That cluster of cells that began to beat in the third week of development did not know what it was starting, but it has not stopped since. And the more carefully we look at how it works, the more evident it becomes that the heart is not just a part of the body. In a very real sense, it is where the body begins.